Hello everyone, this video is about hypersensitivity and allergy, some immunology related topics, and this particular video is using study materials from EPJ Pathshala. So I would like to express my sincere gratitude to the developers of this study note. Thank you. Allergy and hypersensitivity 1. Our learning outcomes, like we will be having our introduction of allergy and anaphylaxis along with hypersensitivity, immediate versus delayed hypersensitivity, gel and Coombs classification of hypersensitive reactions, type 1 hypersensitivity, general aspects, components, allergens, Th2 cells, IgE, tissue mast cells and blood basophils, TCFC receptor, degranulation process and its mechanism, then mediators of type 1 hypersensitivity along with some other aspects like clinical manifestations of type 1 hypersensitivity, regulation of type 1 hypersensitivity, detection of type 1, therapy or precaution, and along with that a summary. So allergy and hypersensitivity 1. After studying this module, you shall be able to know what is allergy, how is it different from anaphylaxis, what are the hypersensitive reactions, how dysregulated immune system or hypersensitivity affects us, then identify the components and mechanisms that mediate each hypersensitive response, then analyze the underlying beauty of our innate immune system in terms of its regulation. So let's have an introduction. The word allergy originates from Greek word allos, meaning other or different, and ergia means energy or action, in the sense of change in reactivity or capacity to react. The concept of allergy was given by Viennese pediatrician von Ferket in 1906 in a classic article entitled Allergy. According to Ferket, an antigen causes change in reactivity which can be protective, immunity, or harmful, hypersensitivity. Then Richard and Portia proposed a different term, anaphylaxis, in contrast to prophylaxis for the overreaction of immune system. A.F. Koka and R.A. Cook, I'm not sure about the pronunciation of these scientists, proposed a general term hypersensitivity to resolve the contradiction between allergy and anaphylaxis. Hypersensitivity is a general term for inappropriate, deleterious immune response raised against an allergen. Gell and Coombs proposed a classification in 1963 according to which different hypersensitivity reactions can be classified in 4 types. The classification is based on the type of immune branch involved, humoral or cell-mediated, type 1, 2, 3 within humoral branch, and type 4 in cell-mediated branch. 1, 2, 3 in humoral, next one cell-mediated. Time taken to show symptoms like immediate or delayed, type 1, 2, 3 are immediate type and 4 is delayed type hypersensitivity. And immune mechanism, cells and mediator molecules involved like this. So type 1 hypersensitivity is IgE based allergic reaction raised against allergens. It is a humoral immunity based immediate type hypersensitive reaction like asthma. Humoral immunity based. So we mentioned all this. Then type 1 hypersensitivity is IgE based allergic reaction raised against allergens. It is a humoral immunity based immediate type of hypersensitive reaction. For example, asthma. Type 2 hypersensitivity is IgG mediated cytotoxic hypersensitivity. Well, wait a minute. Type 1 hypersensitivity is IgE based allergic reaction raised against allergens. It is a humoral immunity based immediate type hypersensitive reaction. For example, asthma. Then type 2 hypersensitivity is IgG mediated cytotoxic hypersensitivity. which is also humoral immunity based immediate type hypersensitive reaction. For example, erythroblastosis fetalis. Then type 3 hypersensitivity is immune complex mediated humoral immediate type hypersensitivity, for example, rheumatoid arthritis. Type 1 example asthma, type 2 erythroblastosis fetalis, type 3 rheumatoid arthritis. IgE mediated, IgG mediated, immune complex mediated. Next, type 4 hypersensitivity is cell mediated hypersensitivity. It is cell mediated immunity based. That's the difference. Delayed type of hypersensitivity reaction like contact dermatitis, for example. Common contact dermatitis. Okay. And basic components of type 1 hypersensitivity are allergens, immune cell cytokines, IgE, Fc receptors for IgE, and various mediator molecules. The degranulation of basophils and mast cells is the key point of type 1 hypersensitivity. Basic components of type 1 hypersensitivity are allergens, immune cell cytokines, IgE, Fc receptors for IgE, and various mediator molecules. The degranulation of basophils and mast cells is the key point of type 1 hypersensitivity. Then clinical manifestation of type 1 hypersensitivity include systemic anaphylaxis and localized anaphylaxis. We are learning in particular about type 1 hypersensitivity. So, type 1 hypersensitivity include systemic anaphylaxis and localized anaphylaxis like allergic dermatitis, hay fever, asthma, etc. Various factors regulate type 1 hypersensitivity response such as dose and type of allergen, processing of allergen, genetic constitution of the individual, and the relative ratio of Th1 and Th2 cells. And wheel and flare test, radioimmunosorbent assay, RIST, and radioallergosorbent assay, RAST. Wheel and flare test, radioimmunosorbent assay, RIST, and radioallergosorbent assay, RAST, are common medical tests to detect the type 1 hypersensitive individuals for a particular allergen. The treatment or precaution involves avoidance of allergen, modulation of immunological response, stabilization of the triggering cell, antagonism of mediators, and attacking chronic inflammation. Various drugs are available to cure type 1 hypersensitive reactions. Concept of allergy and anaphylaxis. The allergy from Greek word allos meaning other or different, and erga means energy or action in the sense of changing in reactivity or capacity to react is a fundamental aspect of clinical immunology. The concept of allergy was given by Venus pediatrician Clemens von Freiherr in 1906 in a classic article entitled Allergy. According to the original idea of von Purkit, when an individual comes in contact with an antigen like pollen, germs, food particle, etc., a change in reactivity occurs. This change, which was termed as allergy by von Purkit, can induce a protective or harmful response. The protective response makes the individual immune to those antigens and hence are called immunity, which does not manifest any symptoms. On the other hand, there is a harmful response which shows symptoms or signs of contact with antigen and is called as hypersensitivity. The immunity and hypersensitivity can overlap and are result of some physiological response, like response of host immune system. A diagrammatic depiction of this idea is represented, which we will be, gazing at. This idea of allergy on right. Antigen, allergy, hypersensitivity, immunity. Unfortunately, this new idea was not easily acceptable due to existing strength of idea of immunity. The revolutionary thought of Purkit came at a time when immunology was establishing as science. The idea of immunity by notable scientists such as Pasteur, Ehrlich, Mechnikov, Bordet, and Bering, etc., in 19th century hinted and delayed the acceptance of von Purkit's idea. Nobody could imagine at that time that immune system can harm the host it protects. The French physiologist Charles Richet in 1850 to 1935 and Paul Poitier in 1866 to 1962 rejected the term allergy as they regarded it unnecessary new term in addition to anaphylaxis that they already coined. These two scientists were suggested by Prince to study the toxic properties of Physalia, Portuguese man of war's jellyfish, in the South Sea in the laboratory abroad the asked Princess Alice too. So Richard and Poitier continued their studies in Paris, but due to unavailability of Physalia, they used Acinia. They experimented with the use of isolated jellyfish toxins as vaccines. They injected dogs with the purified toxins followed by a booster. Instead of producing antibodies against the toxin, the dogs immediately showed vomiting, diarrhea, anaphylaxis, and even death. These dogs clearly overreacted to the antigen, which was termed as anaphylaxis by Poitier and Richet, and contrasts to prophylaxis to describe its overreaction. So Richet was awarded the Nobel Prize in Physiology or Medicine in 1913 in recognition of his work on anaphylaxis. Von Purkit and Richet's perspectives were different, but since anaphylaxis was easier to understand, it gained more interest. The phenomenon of allergy was much broader about the functioning of immune system and its involvement in diseases than anaphylaxis, an initial purely experimental phenomenon. During the research for active substances of anaphylaxis, it was realized that it is not only the effect of toxin, but some principle in the organism itself is also involved. Like Sir Henry Dale, 1875 to 1968, had made a contribution in the elucidation of the same. He, along with co-workers P.P. Elidelo, studied the physiological aspects of histamine and developed the concept of histamine shock as basis of anaphylaxis. I would like to see Clemens von Brigitte Freiherr. This is he. Okay. Charles Richet, Paul Poitier, Sir Henry Dale. Yeah, I'm sorry. Arthur F. Cook and Robert A. Cook published many articles in 1923 to 1926 to resolve the allergy and anaphylaxis contradictions. They preferred to employ the term hypersensitivities as a general term. They divided hypersensitivity to normal type like contact dermatitis, serum sickness, etc., and abnormal type like anaphylaxis, atopy, etc. Later, medical community began to use allergy for human hypersensitivity reactions and anaphylaxis for experimental animals. There were no significant modifications in understanding the idea of allergy or its use until 1963 with Philip George Hortham-Gell in 1940 to 2001 and Robert Royston Amos, Robin Coombs, Jelen Coombs in 1923 to 2006 classified hypersensitive reactions in their book Clinical Aspects of Immunology. Jell and Coombs. Jell and Coombs. So now we are gonna once again read about hypersensitivity. The primary aim of an immune response is to remove the exogenous or endogenous antigen by various mechanisms using effector molecule. The body homeostasis is regained after localized inflammation and removal of antigen without causing a damage to host tissue. So we were learning about this. The primary aim of an immune response is to remove the exogenous or endogenous antigen by various mechanisms using effector molecules. The body homeostasis is regained after a localized inflammation and removal of antigen without causing a damage to host tissue. But under certain circumstances, this beneficial inflammatory response can cause deleterious outcomes, including host tissue injury, disease, or even death. Such a harmful, inappropriate immune response is termed as hypersensitive response or reaction, or simply hypersensitivity. The term hypersensitive implies that a person is excessively or more sensitive to an antigen as compared to a normal response shown by other persons. So it indicates towards an elevated or increased immune response, but it is not the case always. An inappropriate immune response, I'm sorry, an inappropriate immune response is also considered as hypersensitivity besides a heightened immune response. So this is the flow chart. A generalized view to represent protective immune response and deleterious hypersensitive response source. Response. I'm sorry. Normal healthy individual exposure to exogenous or endogenous antigens. Two kinds of generation to immune response is there. Kind protective immune response is there, along with inappropriate increased deleterious immune response is there, which can be referred to as hypersensitivity. But along with that, protective immune response is also there, which helps to regain the body homeostasis, helps which is helping in the regain of body homeostasis. So that is how these ideas are connected. Hypersensitivity is also caused by the exposure of exogenous or endogenous antigens, but that is a kind of inappropriate or increased deleterious immune response. Okay. The immediate versus delayed hypersensitivity. You might have already learned this. Based on the time taken to manifest the symptom, the hypersensitive reaction can be classified as immediate or delayed type. If the symptoms are observed within minutes or hours of antigen encounter in a synthesized recipient. Wait a minute. Based on the time taken to manifest the symptom, a hypersensitive reaction can be classified as immediate or delayed type. If the symptoms are observed within minutes or hours of antigen encounter in a sensitized recipient, the hypersensitive reaction is called as immediate hypersensitivity. The anaphylactic reactions initiated by antibody or antigen-antibody complex like immune complex, humoral immunity come under this category. Delayed type hypersensitivity, in contrast, named so because of delay in appearance of symptoms until several days after the exposure. Hypersensitive reactions in the cause of cell-mediated immune response comes under this category. So, Gell and Coombs classification of hypersensitive reaction. This is a very important topic. So, I would like to slowly explain this because I'm kind of sick, but still I have got an exam, so I need to study this. Based on the type of immune response involved, humoral or cell-mediated, and the effector molecules involved, different immune mechanisms are involved in generation of hypersensitive reactions. Accordingly, hypersensitive reaction can be categorized in different types. The British immunologist Philip George Gell, 1914 to 2001, and Robert Royston Amos Robin Coombs, 1921 to 2006, proposed a classification scheme for hypersensitive reactions in their book Clinical Aspects of Immunology in 1963. They divided hypersensitive reactions as type 1, type 2, type 3, and type 4 hypersensitive reactions. The classification is based on the type of immune branch involved, humoral or cell-mediated, type 1, 2, 3 with humoral branch, and type 4 in cell-mediated branch. Time taken to show symptoms, immediate or delayed. 1, 2, 3 are immediate type and type 4 is delayed type hypersensitivity. So, immune mechanism, cells, and mediator molecules involved. So, here we could see a pictorial representation of different hypersensitive reactions, and this is a kind of complex, but still we will be slowly reading. Okay, type 1. We mentioned earlier that's IgE mediated hypersensitivity. So, antigen specific IgE, allergen, Fc receptor for IgE, degranulation process is also involved there. So, what is happening in this case? IgE mediated hypersensitivity. IgE is bound to mast cells via its Fc portion. So, IgE is bound to mast cells via its Fc receptor portion. When an allergen binds to these antibodies, cross-linking of IgE induces degranulation, causes localized and systemic anaphylaxis, seasonal allergies including hay fever, food allergies such as those to shellfish and peanuts, ice and eczema. Then type 2 antibody dependent cellular cytotoxicity. Cytotoxic T cell Fc receptor for IgG, surface antigen target cell. Complement activation, surface antigen immune complex. Type 2 IgG mediated cytotoxic hypersensitivity. Cells are destroyed by bound antibody either by activation of complement or by a cytotoxic T cell with an Fc receptor for the antibody. Antibody dependent cell mediated cytotoxicity. So IgG mediated cytotoxic hypersensitivity. Cells are destroyed by bound antibody either by activation of complement or by a cytotoxic T cell with an Fc receptor for the antibody ADCC mechanism. Red blood cells destroyed by complement and antibody during a transfusion of mismatched blood type or during erythroblastosis fetalis. Red blood cells destroyed by complement and antibody during a transfusion of mismatched blood type or during erythroblastosis fetalis. Then free floating immune complex, complement activation, neutrophil. Type 3 immune complex mediated hypersensitivity antigen antibody complexes are deposited in tissues causing activation of complement which attracts neutrophils to the site. And most common forms of immune complex diseases are seen in glomerulonephritis, rheumatoid arthritis, and systemic lupus erythematosus. Then the fourth one, sensitized Th cell, antigen, cytokines, cytotoxic cell, activated macrophage like that. So cell-mediated hypersensitivity. Th1 cells secrete cytokines which activate macrophages and cytotoxic T cells and can cause macrophage accumulation at the site. Most common forms are contact dermatitis, tuberculin reaction, along with autoimmune diseases such as diabetes mellitus type 1, multiple sclerosis, and rheumatoid arthritis. Type 1 hypersensitivity. Type 1 hypersensitivity, general aspects, is an immediate type hypersensitivity within the humoral branch of immunity. It is induced by antigens called the antigens or allergens. Type 1 hypersensitivity is similar to normal humoral immune response except the fact that plasma cells secrete IgE antibody in response to activation of allergen specific Th2 cells. These IgE antibodies bind with high affinity to Fc receptors on blood basophils and tissue mast cells. These IgE coated mast cells and basophils are called as sensitized cells. The exposure of some allergen later causes cross-linkage of these membrane-bound IgE on sensitized mast cells and basophils, causing their degranulation. These granules release pharmacologically active mediator molecules like histamines, which primarily cause vasodilation and smooth muscle contraction in a localized manner or systemically. Degranulation will be happening. These granules release pharmacologically active mediator molecules like histamines, which primarily cause. Fc receptors are situated on the blood basophils and tissue mast cells also. These IgE coated mast cells and basophils are called as sensitized cells. The exposure of some allergen later causes cross-linkage of these membrane-bound IgE on sensitized mast cells and basophils, causing their degranulation. These granules release pharmacologically active mediator molecules like histamines, which primarily cause vasodilation and smooth muscle contraction in a localized manner or systemically. The clinical manifestation of type 1 hypersensitivity includes systemic anaphylaxis or localized anaphylaxis including hay fever, hives, eczema, asthma, and food allergy. A generalized type 1 reaction is shown. Yep, here. First, we will be learning the sensitization phase. Allergen is there, which binds. B cell is there. CD4 cell. CD4, then IL4, Th2 cells, memory cell, plasma cell, which produces some allergen specific IgE, B cell, allergen, CD4, IL4, Th2 cell, memory cell, plasma cell, allergen specific IgE, then IgE, sensitized mast cell, Fc receptor for IgE, granules. This is happening in case of sensitization phase. Degranulation phase, degranulation, allergen, vasoactive amines. Degranulation will be taking place. Then effect on surrounding tissue, smooth muscles, blood vessels, nerve endings, etc. The generalized mechanism of type 1 hypersensitive reaction, sensitization phase, involves exposure to allergen, activation of Th2 cells, release of IgE from B cells, and binding of these IgE with Fc receptor of mast cells. Degranulation phase involves release of mediators on Degranulation phase involves release of mediators on re-exposure of same allergen and their further effects on surrounding tissue. Then components. Allergens. The term allergen refers to non-parasitic environment agents which stimulate type 1 hypersensitive response in allergic atopic individuals on repeated exposure. The term allergens refers to non-parasitic environmental antigens which stimulate type 1 hypersensitive responses in allergen atopic individuals on repeated exposure. The significantly high serum IgE levels is mount in human against parasitic infections only. Wait a minute, what is that? The term allergen refers to non-parasitic environmental antigens which stimulate type 1 hypersensitive responses in allergic or atopic individuals on repeated exposure. The significantly high serum IgE level is mounted in people and against parasitic infections only. But the people who are genetically predisposed to development of IgE mediated immediate hypersensitivity or type 1 against common environmental antigens have this abnormality called the atopy. These individuals have regular defects for IgE, because of which non-parasitic antigens also stimulate IgE production, causing tissue damage and hypersensitivity. The inherited atopy phenomenon is multigenic. The genetic cause behind the phenomenon of atopy has been mapped to various loci by linkage analysis, which are on chromosome 5 is present a locus which is linked to a region that codes cytokines IL-3, IL-4, IL-5, IL-9 and IL-13. So granulocyte monocyte colony stimulating factor GM-CSF, all these cytokines promote IgE production. On chromosome 11 is present another locus which is linked to a region that encodes the beta chain of high affinity IgE receptor. And on chromosome 6 is present the MHC genes which have also been linked to IgE response. Some of the common allergens are summarized here. Food in the sense eggs, mushrooms, milk, peas, beans, seafood, nuts. Insect products, venom of bees, ants, wasps, cockroach, calyx, dust mites, etc. Then drugs, salicylates, penicillin, anesthetics, sulfonamides, plant pollens, ragweed, ryegrass, birch trees, proteins, vaccines, foreign serum, etc. Others, mold spores, latex, animal hair, etc. Most of the allergens are small sized, soluble in nature, glycosylated proteins or protein bound substances with molecular weight 15,000 to 40,000. The allergenicity of an allergen depends on its ability to evoke a type Th2 mediated immune response, which in turn depends on a number of factors such as type of allergen, dose of allergen, sensitizing route, adjuvant, recipient's genetic or hereditary predisposition. That's kind of atopy. Hallucination. Why do some allergens like ragweed pollens induce potent allergic responses, whereas other equally abundant allergens like needle pollens do not? It depends upon the various factors listed above, such as sensitization routes, adjuvant, and genetic constitution of host, beside type and dose of allergen. Sensitization routes, type of allergen, dose of allergen, sensitizing route, adjuvant, recipient's genetic or hereditary predisposition. Predisposition in the sense atopy. Then Th2 cells. The type of immune response against a particular antigenic challenge is determined by the cytokine secretion pattern of different subtypes of Th cells. CD4 plus Th cells exert their functions to cytokines in autocrine or paracrine way, whereas there are two subtypes of CD4 plus. Wait a minute. Yeah, thank you for your patience. So Th cells exert their functions through cytokines in autocrine or paracrine way. There are two subtypes of CD4 plus Th cells, Th1 and Th2 cells. These two subsets differ in the profile of cytokines they secrete. Th2 cells secrete GM-CSF, IL-3, IL-4, IL-5, IL-10 and IL-13. The primary functions of Th2 cells include IgG production, eosinophil and mast cell activation and differentiation, and help to B cells. So IL-4 secreted by Th2 cells promote the class switching in B cells from IgM to IgE. The effect on IgE production meshes with the differentiation and activation of eosinophil by IL-5 because eosinophil possesses Fc gamma receptor. No, gamma, it's not gamma. It's epsilon, Fc epsilon receptors which bind IgE. The parasitic infections and allergic reactions, they induce Th2 response leading to anti-parasitic and type 1 hypersensitive reactions. The Th2 cytokine profile is higher in allergic diseases and helminthic infections. IgE. Immunoglobulin E is named so in reference to E antigen of ragweed pollen, which is a potent inducer of this class of antibodies. Though the amount of IgE in serum is very low, but its potent biological activity made it to be identified. Atopic individuals have 10 times more serum IgE level than normal ones, so that can be an indication one person has atopy. So it was identified by Otto called Willi Brustinus, I'm not sure about the pronunciation, 1876 to 1963, and Heinz Kestner in 1897 to 1963 in 1921 as a serum component responsible for allergic reaction. They injected serum from an allergic person to a non-allergic person intradermally. The later injection of appropriate antigen at the same site caused swelling and redness termed as wheel and flare reaction. This reaction is called as PK reaction after the name of their discovered Brusnitz and Kestner. It was the first evidence of biological activity of IgE, though the IgE is not known at that time. The IgE was first identified by Kimmich and Teruga Ishizaka in 1966. They immunized the rabbit with serum of an allergic human individual to prepare anti-isotype antiserum. The serum of rabbit thus obtained was reacted with all the known antibodies at the same time, IgA, M, IgD, IgA. This way, each of the known anti-isotype antibodies was precipitated and removed from the rabbit antiserum. The remaining serum was still having an antibody which could block the PK reaction. This new class of antibody was named as IgE. IgE mediates two immune functions like allergic reaction or type 1 hypersensitive reactions and antibody dependent cell mediated cytotoxicity or ADCC, anti-parasitic defense. Then IgE has higher molecular weight than IgG due to additional constant region domain which replaces the hinge region of IgG. Wait a minute, IgE mediates two immune functions, which are the allergic reaction or type 1 hypersensitive reactions we mentioned, and an antibody dependent cell mediated cytotoxicity, anti-parasitic defense, which we will be shortly representing with the help of this ADCC. Okay, ADCC and type 1 hypersensitivity. And IgE is a kind of heavy in the sense is of higher molecular weight due to additional constant region domain which replaces the hinge region of IgG. Here we could see these two scientists, PK reaction, wheel and flare response, I guess. Yeah, that's what it is called. Yeah, same thing. Properties of human serum immunoglobulin E, IgE. Molecular weight, heavy chain component epsilon, light chain component kappa or lambda. Molecular formula given, normal serum level, in vivo serum half-life days 2.5 days, Primary function mast cell and eosinophil degranulation. Mast cell and eosinophil degranulation. Tissue mast cells and blood basophils. Mast cells originate from CD34 plus, CD117 plus, CD13 multipotent hematopoietic progenitor cells in humans in bone marrow, from where they migrate to various tissues through blood where they mature. So they were first identified by Dr. von Recklinghausen. I'm not sure of the pronunciation, just read this name. In 1863 as granular cells in the mesentery of the frog. Initially, mast cells were termed or named as Mastzellen by Dr. Paul Ehrlich in 1878, also known as mastocyte or labrocyte. It was the discovery of histamine in 1910. Slow reaction substances of anaphylaxis, now leukotrienes, in 1938 and IgE in 1966 that provided initial insights into the role of mast cells in allergic reactions. A number of stimuli can activate mast cells, which can then release a spectrum of mediators in discriminating manner. These mediators include histamine, proteases and other enzymes, growth factors, chemokines, cytokines, reactive oxygen and nitrogen species, and arachidonic acid metabolites. These mediators perform their roles in allergic reactions. So mast cells adapt to various phenotypes based on their microenvironment. This property is called as mast cell heterogeneity. A number of stimuli can activate mast cells, which then release a spectrum of mediators in discriminating manner. These mediators include histamine, proteases and other enzymes, growth factors, cytokines, chemokines, reactive oxygen and nitrogen species, and arachidonic acid metabolites. These mediators perform their roles in allergic reactions. Mast cells adapt to various phenotypes based on their microenvironment. This property is called the mast cell heterogeneity. Mast cell heterogeneity is decided on the basis of their location, content of protease, histochemical staining, and reactivity to anti-allergic drugs. Then rodents have two major subtypes of mast cells, like mucosal type and connective tissue type, like MCT and MCTC. Whereas humans have the three major subtypes of mast cells on the basis of content of serine proteases, like tryptase only type MCT, chymase only type MCC, or both tryptase and chymase positive type MCTC. The primary functions that mast cells perform are maintenance of homeostasis, act as interface between innate and adaptive immunity, protective roles in various infections, role in allergic or type 1 hypersensitive reactions. Here you could see a picture of a cultured mast cells in 1000x magnification stained with tool blue. Difference between mucosal and connective tissue mast cell. T cell dependence present in case of mucosal mast cell, but it is absent in case of connective tissue mast cell. Histamine release and degranulation is kind of higher in case of connective tissue mast cells. Degranulation blockage by disodium chromoglycate theophylline is present in case of connective tissue mast cell, absent in case of mucosal mast cell. Basophils. Basophils are the non-phagocytic granulocyte leukocyte that are found in most vertebrates. They are the least abundant population of granulocyte in peripheral blood, comprising not 0.5 to 1% of total leukocytes. Non-phagocytic granulocytic leukocytes that are found in most vertebrates. And the granulated cytoplasm of basophils stains with basic dye, and hence the name basophils. They were discovered by Paul Ehrlich in 1879 based on their unique microscope appearance on exposure to basic dyes. Basophil develops from basophil precursor cells which in turn originate from hematopoietic stem cells or HSCs, derived granulocyte monocyte progenitor GMP cells in bone marrow. They have a short lifespan of around 1 to 2 days long only. They contain a multi-lobed nucleus, some mitochondria, glycogen granules and membrane-bound granules containing pharmacologically active mediator molecules. The primary functions is release of histamine and leukotrienes in response to IgE-mediated activation, role in type 1 hypersensitivity. Basophils have roles in inflammation and associated diseases such as urticaria, asthma, etc. Also, they are involved in protection against parasitic infections. We could see a basophil along with RBC and a diagrammatic view of a single basophil. And FC receptors. The biological activity of IgE depends upon its binding with its receptors. These receptors are specific for the FC region of a epsilon heavy chain of IgE antibody. Two types of FC epsilon receptors are there based on their difference in affinity for IgE, cellular distribution and their effects post IgE binding, like FC epsilon R1 and FC epsilon R2 or CD23. Two types of FC epsilon receptors are there based on the difference in affinity for IgE, cellular distribution and their effects post IgE binding, FC epsilon R1 and FC epsilon R2. FC epsilon R1 also called as high affinity IgE receptor because of its capacity to bind IgE despite low serum concentration of IgE, 1 into 10 raised to minus 7. A single human basophil contains this much of FC epsilon R1. FC epsilon R1 is present primarily on cells such as mast cells, basophils, Langerhans cells, monocytes. Structurally, the FC epsilon R1 is made up of 4 polypeptide chains, 1 alpha, 1 beta and 2 gamma chains. Alpha chain. The amino terminal of alpha chain lies in extracellular region whereas carboxylic 1 lies in cytoplasm. The extracellular region contains 290 amino acid domains which resemble immunoglobulin fold structure. These immunoglobulin fold-like structures bind with CH3, CH3 and CH4, CH4 domains of IgE. And beta chains. The beta chains has its amino acid carboxylate terminal in cytoplasm only. It spans the plasma membrane 4 times. It acts as linker between alpha and gamma chains. Then del gamma chain. Two identical gamma chains are there which are linked together by disulfide linkage. Each gamma chain possesses a conserved sequence called as immunoreceptor tyrosine based activation motif ITAM. Immunoreceptor tyrosine based activation motif. Immunoreceptor tyrosine based activation motif. Immunoreceptor tyrosine based activation motif. Immunoreceptor tyrosine based activation motif ITAM in its cytoplasmic domain. This ITAM motif interacts with tyrosine kinase to transduce an activation signal to cell. And the allergens upon binding to IgE cause cross linkage leading to aggregation of FC epsilon R1 receptors. Immunoreceptor tyrosine based activation motif interacts with tyrosine kinase which further causes tyrosine phosphorylation and mast cell degranulation. Ig-like domains, extracellular space, plasma membrane, cytoplasm, COH. This high affinity of IgE receptor FC epsilon R1. And FC epsilon R2, also called the low affinity IgE receptor, also called CD23, specific for CH3 domain of IgE. FCR2 is present primarily on cells such as B cells, eosinophils, alveolar macrophages. Structurally, it is composed of a single polypeptide chain with its carboxylic terminal in extracellular region and amino terminal in cytoplasm. The extracellular domain contains disulfide linkages. The functions of Fc epsilon R2 include IgE transport across human intestinal epithelium, positive feedback mechanism to enhance allergic sensitization, B cells, activation of alveolar macrophages and eosinophils. Fc epsilon R2 receptor, degranulation process and its mechanism. Degranulation is simply a cellular process of releasing bioactive molecules from cytoplasmic granules or secretory vesicles in cells like mast cells and granulocytes. With respect to type 1 hypersensitivity, we will discuss the degranulation of mast cells and basophils primarily. So both these cell types show similar mechanism of degranulation with minor differences. For simplicity, let us discuss the general mechanism of degranulation irrespective of cell type involved first. So the binding of allergen to bound IgE on cell surface doesn't trigger the degranulation process. The initiation of degranulation requires cross-linking of IgE receptors present on these cell surfaces. The significance of cross-linkage lies in the fact that the monovalent allergen cannot initiate or start degranulation because of its inability to cross-link the bound IgE receptors. The experimental evidence has proved that IgE to allergen ratio of 2:1 or more can only induce degranulation. Different mechanisms can cross-link this IgE receptors. IgE allergen ratio lesser than this could not induce degranulation due to its inability of cross-linking. Then another important observation that emerged from these experiments was that the cross-linkage of 2 or more Fc epsilon R1 is required for degranulation initiation with or without bound IgE. Various mechanisms, including the ones that can bypass the need of allergen or IgE, are shown in the figure. Normal mechanisms like cross-linkage of cell-bound IgE by allergen. Wait a minute. Normal mechanism, cross-linkage of cell-bound IgE by allergen. Mechanisms that do not require antigen, cross-linkage of cell-bound IgE by anti-isotype or anti-idiotype antibody. Cross-linkage of cell-bound IgE by allergen. Cross-linkage of cell-bound IgE by anti-isotype or anti-idiotype antibody. Then cross-linkage of cell-bound IgE by chemical. Then mechanisms require neither allergen nor IgE. Cross-linkage of cell-bound IgE by anti-receptor antibody. Mechanisms require neither allergen nor IgE and not even cross-linkage of receptor. Calcium ion influx by ionophore and increased permeability for calcium. Cross-linkage of cell-bound IgE by allergen IgE, Fc receptor IgE, mast cell. The next cross-linkage of cell-bound IgE by anti-isotype and anti-idiotype antibody. Anti-idiotype AB, IgE, mast cell, anti-idiotype AB, IgE, mast cell. And cross-linkage of cell-bound IgE by chemical. Cross-linkage by IgE by chemical, mast cell. Possible mechanisms which can trigger the initiation of mast cell degranulation. Biochemical events in activation and degranulation of mast cells. A series of well-coordinated intracellular events helps in activation and degranulation or release of mediators from mast cells like basophils and various proteins and lipid kinase and phosphatase along with cytoskeletal rearrangements are involved in these biochemical events. A sequential mechanism along with a pictorial depiction of the same is elaborated. Allergen cross-linking of bound IgE or by other mechanisms listed above, aggregation of Fc epsilon R1, activation of protein tyrosine kinase PKTs present on cytoplasmic domains of beta and gamma chains of Fc epsilon R1, PKTs phosphorylate tyrosines with ITAMs of gamma subunit of Fc epsilon R1 resides on beta subunit of Fc epsilon R1 and phospholipase C. Phospholipase C converts phosphatidyl inositol 4,5-biphosphate PIP2 into diacyl glycerol DAG and inositol triphosphate IP3. Then DAG activates protein kinase C PKC, which is necessary for microtubular assembly and fusion of granules with plasma membrane along with Ca2+. IP3 on the other hand is responsible for intracellular mobilization of Ca2+. Ac epsilon R1 cross linkage also activates other enzymes that convert phosphatidylserine into phosphatidylinositol. Lamine. P is methylated to phosphatidylcholine by phospholipid methyltransferase enzyme 1 and 2, PMT1 and 2. PC accumulation on exterior surface of plasma membrane cause an increase in Ca2 plus influx. Then Ca2 plus influx activates phospholipase A2 that breaks down PC into lysophosphatidylcholine and arachidonic acid. Arachidonic acid is converted to leukotrienes and prostaglandin D2 and mediators, the mediators. Then Fc epsilon R1 cross linkages also activate the enzyme adenylate cyclase, which causes a constant increase in CAMP within 15 seconds. Later, CAMP is stopped by protein kinase PKC. Protein kinase phosphorylate to the membrane proteins of granules that further changes the permeability of granules for water and Ca2+. The resulted swelling in granules facilitate their fusion with plasma membrane and ultimately the degranulation release of mediators. Mediators of type 1 hypersensitivity. Mediator molecules are pharmacologically active agents that are released from the mast cells and basophils in the process of degranulation. It is the mediators which are responsible for biological effects or clinical manifestation of type 1 hypersensitivity. They act as amplifying terminal effector mechanisms which act upon local tissues and secondary effector cells, neutrophils, eosinophils, lymphocytes and platelets, etc. These mediators for mast cells and basophils have a protective defensive role against parasitic infections where they cause vascular permeability and vasodilation to bring an influx of plasma and memory inflammatory cells to attack the pathogen. But the same phenomenon in case of an inappropriate non-pathogenic allergenic induction causes deleterious effects such as in the case of type 1 sensitive hypersensitive reactions. Based on the synthesis and storage, mediators are classified primary or secondary mediators, shown here with their biological activity. Primary mediators, they are pre-manufactured mediators which remain stored in granules of mast cells and basophils. Histamine, serotonin, heparin, proteases are some of the common primary mediators which are listed below along with their biological roles. Heparin, histamine, smooth muscle contraction and increase in vascular permeability. Serotonin, smooth muscle contraction and increase in vascular permeability. Neutrophil chemotactic factor or NCFA, that is something which helps in the chemotaxis of neutrophils. Then eosinophil chemotactic factor or ECFA, chemotaxis of eosinophils. Then protease, tryptase and kinase, bronchial mucus secretion, generation of complement split products, degradation of blood vessel basement membrane. Secondary mediators, they are not preformed like primary mediators but are either synthesized upon target cell activation or released upon breakdown of membrane phospholipids during the process of degranulation. Some of the common secondary mediators are leukotrienes, prostaglandins, cytokines like IL-1, IL-4, IL-5, IL-6, TNF-alpha, etc. and platelet activating factor. Their biological functions are listed. Leukotrienes, contraction of pulmonary smooth muscles and increased vascular permeability, also called a slow reactive substance of anaphylaxis, SRSA. Then prostaglandins, contraction of pulmonary smooth muscles, platelet aggregation, vasodilation. Chemokines, IL-1 and TNF-alpha, systemic anaphylaxis, IL-4 and IL-13, IgE production increase, IL-5, eosinophil activation and generation. Then bradykinin, contraction of smooth muscles and increased vascular permeability. Then platelet activating factor, contraction of pulmonary smooth muscles, platelet aggregation. Some of the common secondary mediators molecules with their biological roles. The difference observed in clinical manifestations of type 1 hypersensitive reactions in different tissues or different species can be different because of variation in primary and secondary mediators. I am running out of time, so we will be quickly reading this. Clinical manifestations of type 1 hypersensitivity. The clinical manifestations of type 1 hypersensitive reactions are classified as localized anaphylaxis like eczema, asthma, food allergy and rhinitis or hay fever, or systemic anaphylaxis like depending on the extent of tissue organ damage whether it's localized the target site or systemic. Various type 1 hypersensitivity clinical manifestations are like systemic anaphylaxis. It is a shock-like fatal condition which involves throughout body symptoms. Its symptoms are observed within minutes of type 1 hypersensitive reactions. Common allergens which can cause systemic anaphylaxis in susceptible individuals are venom of bees, wasps, and ants, drugs such as antitoxin, penicillin, penicillin, food such as nuts and seafood, etc. A widely used drug for treatment of systemic anaphylaxis is epinephrine. Epinephrine reverts back the effects caused by mediators. The reduced vascular permeability and relaxes the smooth muscles, blocks mast cell degranulation by increasing CAMP level and prevent vascular collapse by improving cardiac output. Guinea pig is the preferred animal model for studying systemic anaphylaxis. Then localized anaphylaxis is also there. Localized in the sense, in this response, reaction is restricted to specific target tissue or organ, only usually epithelial tissue at the site of allergen entry. It is also called an atopy, the inherited tendency to manifest localized anaphylaxis. Common example of localized anaphylaxis are allergic rhinitis, hay fever, atopic dermatitis or allergic eczema, food allergies and asthma. Allergic rhinitis is caused by reaction of airborne allergens with sensitized mast cells in nasal mucosa and conjunctiva. Acetylation and increased vascular permeability result due to the mediators which cause sneezing and coughing, watery conjunctiva and nasal mucosa. Callus muscle, smooth muscle, lumen mucus secretion. Increased mucus secretion, narrowed lumen, thickened smooth muscle, trachealis. Hay fever, normal nose with thin watery discharge. Runny nose with thin watery discharge. Runny nose with watery to thick yellow discharge. Fever, no, yes. Sneezing, more common than cold, less than hay fever. Immediate on exposure to allergen, 1 to 3 days after exposure to cold virus. Lasts till the exposure to allergen, 5 to 7 days. Atopic dermatitis or allergic eczema. Erythematosus and pus-filled skin eruptions generally seen in atopic infants are another example of localized anaphylaxis. Infants affected in face, elbows and knees. Allergic eczema, atopic dermatitis in infants. Then food allergies. Susceptible individuals to localized anaphylaxis against a number of food items. Depending upon the site of allergen entry, symptoms may include diarrhea and vomiting in gastrointestinal tract. Asthmatic attacks in respiratory tract or atopic urticaria or erythematosus and edematous eruptions in skin. So hives of urticaria, urticaria. I'm sorry for mispronouncing this urticaria. That's a plant. This is urticaria developed on the skin as an outcome of allergy to food allergen. Asthma can be intrinsic, allergen independent, generally cold or exercise induced. Then allergen dependent, induced by dust, fumes, pollen, insects, etc. In asthma, the target site is lower respiratory tract. Bronchoconstriction and airway obstruction are common features of asthmatic patients. Asthmatic response is divided into early and late phase response. Early response include leukotrienes, prostaglandins and histamine which cause vasodilation, bronchoconstriction and mucosecretion. Later phase involve mediators like IL-4, IL-5, TNF alpha, platelet activating factor, etc., which recruit eosinophils and neutrophils. These inflammatory cells cause tissue damage by releasing toxic enzymes, cytokines, and free radicals. These effects cause accumulation of thick mucus and cell debris which blocks the airway. Hypertrophy of bronchial smooth muscles, thickening of basement membrane, and formation of mucus plugs are other effects observed in asthmatic patients. Cushman spirals are the mucus plug containing clusters of detached epithelial cells and inflammatory cells and spirals of bronchial tissue. They are named after his discoverer. Normal airway during asthmatic symptoms, narrowed airway, limited airflow, tightened muscles, constricted away, blood airway wall, inflammatory thickened wall. Next is about regulation of type 1 hypersensitivity, which we will be learning with the help of next video. So thank you for listening. In that video, we will be learning some aspects of this regulation of type 1 hypersensitivity like allergen dose, genetic constitution of the individual, um, some other points like that, mode of allergen presentation, relative level of Th1 and Th2, then wheel and flare test in, yeah, detection mechanisms. Thank you.